In this video, we will discuss about one of the recently low-cost sequencing technique that has been developed. That is Tequila Seq. So, Tequila is the acronym, and its full name is Transcript Enrichment and Quantification Utilizing Isothermally Linearly Amplified Probes in Conjunction with Long-Read Sequencing. So, this is the full name of this Tequila Seq. So, let us quickly go through the overflow of this Tequila sequencing, how it is Tequila sequencing carried out. So first we have the target gene. So what the initial step they do is they prepare a oligo of this, they prepare a oligo pool of the target gene with a universal adapter, that is universal 3' adapter. After that they use this DNA polymerase and this nicking nuclease to form a biotinylated probes, and this is the step that is cost effective in this Tequila Seq process. So first they form the oligo pools of the target gene, then they form a biotinylated probe with the help of this nicking nuclease and this strand displacement strategy. After that, when they have this biotinylated probe of the target gene, what they do is they do the reverse amplification of the mRNA to form reverse transcription, and PCR amplification to form the complementary cDNA of the whole mRNA. And after that they purify the complementary DNA. They purify the complementary DNA with the help of beads, so that unbind probes will be washed away. Unbind probe will be washed away, and only the biotinylated probe or the probes that take place into this complementary DNA formation will be filtered out with the help of the beads that they have used. This is the step that is shown here, that is capture, wash and amplify. So in this, whatever the excess biotinylated probes are there, they will be removed and washed away. After that they further amplify this complementary DNA with the help of this nanopore sequencing, and this helps them to amplify the complementary DNA that has been formed in this during reverse transcription and PCR amplification. And lastly, they also compared the results that they observed from this Tequila Seq and from the well-established sequencing strategy, that is XGen lockdown, and the results are quite comparable. In this case, we can see that the enrichment in the Tequila Seq is also comparable to the XGen lockdown, that is 84 and 86%. And also the top 30 enriched gene are also in line with the top 30 enriched gene from XGen lockdown. And this is the control experiment in which they check where they don't have the target gene. In that case, whether they get the target results or not. And we can see that in the control experiment where they don't have any target gene, they do not see any target results. So the chances of false negative is very low in this case of Tequila Seq compared to the in case of Tequila Seq. And this Tequila Seq strategy, Tequila sequencing low cost technique can be used reliably to carry out the sequencing of the target genes required for the experiments. And they, in order to further validate or in order to further support their Tequila Seq enrichment, the paper also did sequencing using this Tequila Seq technique on the cancer cells, and they are able to identify and differentiate the different isoforms that are abundant in the case of cancer cells, or more specifically in the case of breast cancers that they have done the experiment with. If you need to understand more about this Tequila Seq uh, low cost sequencing technique, then you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.